Hello to everyone, I am Professor Masood Fuzail. Today we are going to discuss gram staining. Gram staining is a procedure which is introduced by Christian Graham. This procedure is used to identify different groups of bacteria. On the basis of gram staining, there are two groups of bacteria which can be identified as gram positive and gram negative bacteria. As you know that bacteria are microorganisms, they are prokaryotes. So let's discuss on this topic. So according to the classification on the basis of gram staining, bacteria can be divided into two groups, gram negative bacteria and gram positive bacteria. So first of all, we are going to look at the structure of both bacteria, gram negative and gram positive. How they are classified, we will discuss later in this lecture. First of all, I am going to take a cross section of a cell wall of gram negative bacteria. Gram negative bacteria has a cell wall outer to their cell membrane. So this is the cell membrane, or you say plasma membrane. As you know that plasma membrane is made up of lipid bilayer. These are lipid molecules which are arranged, and in between the protein molecules are embedded inside the lipid bilayer. Outside the plasma membrane, there is a thin layer of molecule which is known as peptidoglycan. Peptidoglycan is a huge molecule which is known as saccharose. It is made up of fragments of amino acids and a long chain of glycan, which is a carbohydrate. So both together make a molecule which is known as peptidoglycan or murine. Between the peptidoglycan and plasma membrane, there is a space which is known as periplasmic space. In gram negative bacteria, just like inside the cell wall, there is one more membrane outside of the cell wall. This is also a lipid bilayer in which protein molecules are embedded. Some are superficial and some are embedded inside the plasma membrane. These are porous molecules through which molecules can transport, and there are also attached some lipid molecules with this plasma membrane. So there is a very thin layer of cell wall present between these two layers. So because there is a very thin layer of peptidoglycan molecule between the two membranes, that is why we call these bacteria gram negative. But how they call gram negative, we will discuss in the later section of this video. Now we are going to study the cross section of the cell wall of a gram positive bacteria. So let's take a look at this diagram. This is a gram positive bacteria. This is the plasma membrane of bacteria. Plasma membrane is made up of lipid bilayer in which protein molecules are embedded. Just outside towards the plasma membrane, there is a cell wall which is known as peptidoglycan, as you can see in the blue color. So this is a very thick layer of this cell wall, peptidoglycan, as compared to the gram negative. Gram negative bacteria have only very thin layer of peptidoglycan between these two membranes, but gram positive bacteria only has a single membrane, and outer to the plasma membrane there is a thick layer of peptidoglycan molecule which is their cell wall. This peptidoglycan layer can also contain some amount of lipoteichoic acid and teichoic acid molecules. Now, how these two group of bacteria, gram positive and gram negative bacteria, are identified? So first of all, we are going to take bacteria and we are going to put these bacteria on a glass slide. We usually do it in the lab. So this process is known as heat fixation or just fixation. So we take a bacteria and we make a smear, a thin layer of bacteria on the surface of a glass slide. Then we heat up the glass slide so that bacteria can adhere with the glass slide firmly, so that cannot be easily removed. So this process is known as fixation or heat fixation. So you can see these are bacteria which are fixed on the surface of a glass slide. The next step of gram staining is adding a dye which is known as crystal violet dye, which is purple in color. We are going to put a drop of dye which is known as crystal violet on both slides which contain bacteria. Both groups of bacteria will absorb this dye and they become purple and they become violet, as you can see in this diagram. Bacteria usually have no color, so under light microscope when we try to observe them, we are not able to see them. So to observe in the under the light microscope, we have to dye them, we have to stain them. So this is a usually a regular procedure in the biological labs. So after staining with crystal violet dye, then we are going to treat with iodine. So what is the function of iodine? Iodine will make the color of the dye stable with bacterial cell wall. After treatment of iodine, now we are going to remove the color. This process is known as decolorization. This process can be done with alcohol or mixture of alcohols. So we can use any type of ethyl alcohol and we can remove the color. So what happened with the gram and the bacteria which were gram negative bacteria, which had very thin layer of cell wall, very thin layer of peptidoglycan, they will lose their color. Which color? Crystal violet, which we use to stain them. They will lose the color and they will become colorless. Again, it means these bacteria are gram negative, but the procedure has not been finished yet. But the gram positive bacteria, which usually have thick cell wall made up of peptidoglycan, will not lose the color and will still remain violet or purple. Why? Because they had absorbed the crystal violet dye and made a complex which is known as crystal violet complex 1, CV1 complex. So due to the thickness of their cell wall, they cannot be decolorized. So these bacteria are known as gram positive. Now the final step will come. Now we are going to stain again these both bacteria with another dye which is known as safranin. When we add safranin dye, the bacteria which were gram negative, which loses their violet dye and become decolorized, they are going to become pink, reddish pink color. This will confirm that these bacteria are then known as gram negative, but the bacteria which were not be able to decolorize and they will not absorb this safranin dye and still remain violet or purple in color, it means these are gram positive bacteria. So in this way, Christian Graham developed a technique to group or classify the bacteria on the basis of gram stain. Which is also known as crystal violet dye. On the basis of these bacteria has been classified into gram positive and gram negative. I hope this concept will be clear. Hopefully see you in the next lecture. Until then, bye.